ninth meeting of CFA Critical Focus Area Molecular Breeding, and a very warm welcome to all the colleagues sitting here from Patancheru and also our other colleagues from Sub-Saharan Africa who have joined or will be joining very soon in this meeting. So generally in all of our CFA meeting, what we do that we have half an hour discussions related to the different CFA topics, and then we have different seminars. But today we have changed the format little bit because of our guest speaker, eminent speaker, and we thought that today we will have the CFA meeting completely devoted for the presentation. So let me give very brief bio of Dr. Peter Winter. So in terms of the biography, he did his PhD long back in 1986 in the plants, and then from the plant side, from the plants he moved, or he had I think a stint for about 7 years in the human genetics. Is what cancer biology at that time? No, this was genetic mapping. At that time this genetic mapping was just started, and he's having basic background in the area of molecular biology. And then after 1986 to 1993 he was in that human genetic side. Then in 93 he joined the research group of Professor Gunter Kahl at Frankfurt University in Germany, and then he stayed in that group or in that university for about 11 years from 93 to 2004. And in those when he was there, and this is I always tell this thing to different colleagues working on chickpea here at ICRISAT and also at other places. So I think that one of the major or contributions, there are of course Peter has made several contributions, but if you would like to see that which group at international level was responsible for accelerating or not accelerating, initiating the genetics or genomics research in chickpea, then this was the group at Frankfurt University. These were the guys who developed the first set of SSR markers. Even before that in-gel hybridization and lot of technology, and then the first set of the genetic map, for instance. So all those things what they did in the chickpea. So basically I say the chickpea genomics research was initiated from Frankfurt group, where Peter played a very important role, of course, together with Professor Gunter Kahl, and of course at the same time or at later stage there were some other groups in Canada and United States, they also doing the work. And if you see the first SSR map, etc., they are coming from Peter. As you know that we are doing a lot of work right now in the area of molecular breeding, and then we always present that this QTL introgression and the drought tolerance QTL hotspot. So even in this QTL hotspot, the first set of the markers and this classic marker TA170 is coming from Peter, and of course the range of the marker. And then we have been very formal and informal collaboration, and Peter has been associated with ICRISAT, and he has visited ICRISAT several times, especially even when Dr. Jagdish Kumar was here, Jonathan Crouch. When Jonathan Crouch was starting the chickpea genomics research at ICRISAT, he invited Peter and he wanted to have help of Peter to contribute how to start or how to accelerate the chickpea genomics at ICRISAT. I know Peter from my PhD days when I used to work although in wheat, but even at that time as a PhD student I visited Peter and Gunter in Germany, and then after that when I was in Germany I used to have frequent visit to Frankfurt, and when I came to here at ICRISAT we had a lot of interesting discussion. Peter says this is great, now we have a lot of opportunities, and then this group was also very good collaborator with us on many projects including high density genetic maps, and recently completed chickpea genome sequencing, where again Gunter and Peter they played very important role. Right now we are having a project, Indo-German Science Technology Cooperation, IGSTC project, where Peter is representing the industry side and Gunter Kahl is representing academic side, and then where now Hema, Mahender and many other colleagues from ICRISAT we are, and Puran Gaur, so we are the co-PIs on these projects, where we are generating huge amount of sequence data being generated either at Frankfurt or at ICRISAT, and then we are working together on different aspects. So in this project, so then Peter was coming to Delhi and I invited him that this will be a good idea to have some discussion, and today during lunch together with Noel Ellis we had very interesting discussions with Peter. Is that right, Noel? Noel, I think you please come there in the center, and then you can have nice presentation of slides. Nice view of the slides. Yeah. So anyway, so then we had very good interaction, and then we would look forward to have more collaboration, more interactive collaborative project with Peter. And now Peter will be going to present about this work on the okay, sorry, in this I was carried away with my relationship and friendship with Peter, so I forgot to complete his introduction. So, so after working in the area of chickpea, especially devoted completely to chickpea for 11 years till 2004, when I moved to ICRISAT in 2005, Peter thought, now I need to move to some other areas as well because Rajiv can take care of chickpea, right? Anyway, that's a joke. But then he was the founding director or the CEO or manager of this company called GenXPro GmbH. In German we call GmbH. So he is the managing director of GenXPro GmbH company, and this company is very leading company in the area of genomics. Or hey Peter, you had this one-liner, your gene for discovery. What was that? You removed it? The one-liner of genomics. What was that? Yeah, genes never seen before. So they have this one-liner. The GenXPro company can provide you the genes which you have never seen before. So they had the genes never seen before. I used to like that one, so that's what I'm missing. Anyway, so he's working there, or and in his company, now they are taking care of different projects, not only in chickpea, on a range of the plant species. And I think that they are having majority of the work from the human side, from the different hospitals. So now he's having, so basically they are having great expertise in the area of molecular biology. And we always keep on talking, and then this is one of the major emphasis of Peter's presentation today. And Peter and I share, like many of you, the same vision that when we do this kind of research, how to translate this thing in the farmer's field and how to take these things in the nurse field. That's what Peter is going to present, that how to use this huge amount of sequence data to address the issues related to the drought tolerance. And I think that's the reason that I thought that this meeting will be the part of the CFA, because one of the major emphasis of our critical focus area is molecular breeding for the complex trait like drought, etc. And today Peter is going to talk about these things. So with these words, I will not say few, lot of words. I would like to request you to join me in welcoming Dr. Peter Winter. So Peter, floor is yours, and you can come here and then you can start. Manjula, you have that this pointer. I always request you to bring that pointer, but if not, then Peter, you can use this thing. You have the pointer, so bring that pointer. Yeah. Okay. And then I'm going to switch off the light. And then, okay.
Hello.
Okay.
Yeah. Welcome everybody. So you know a lot about me already, and I should say I'm really grateful for this invitation to come to ICRISAT again, and especially to see all the people that have made so impressive progress the last year. And I think it is not just Rajiv, but it is a whole team that really impressed me and actually delighted me to see the progress in chickpea, not only the chickpea genome, but also the many different aspects, for instance, the many populations available, well characterized, well phenotyped. Thank you, Turan, for this. And so I think this is not only the molecular side, but also the breeder side that is very well represented here. And so for me, it is really a big pleasure to be here. So today, I'm going to talk about TranSNiPtomics. That is another omics. And what that is, I will come to that later. But I will show the power of TranSNiPtomics. Profile provided by next generation-based massive analysis of cDNA ends, which we call MACE, which allows you to simultaneously identify allele-specific differential gene expression, in this case of root trait-related drought-response genes, in drought-tolerant and susceptible chickpea varieties. The people that you see listed here are mostly from my lab, but of course from the ICTSAT side, Himabindu Udapa is involved, and of course also Rajiv, and I think another set of other people here that are not mentioned here. I apologize, I do not know you all. This is, Rajiv gave already a short introduction to GenXPro. GenXPro is actually a company that is concerned with next generation based technologies that are applied to many different, almost all aspects of the life sciences. And so what we provide is transcriptomic, genomic, metagenomic, and technologies, wet lab technologies, but also the bioinformatics that is needed to process the large amount of data that are produced by these different technologies. So, and today I will only talk about massive analysis of cDNA, which is a transcription profiling technology. But of course, we provide also a lot of other services concerned, for instance, with the analysis of small and microRNAs, not only in tissues, but also in body fluids and in exosomes, something that's very interesting in the moment. We analyze other non-coding RNAs, we analyze degradomes, and on the genomic level, we discuss that. We also have a technology that allows epigenetic analysis. So these are the machines that we usually use. This is Illumina HiSec 2000, which allows you to sequence approximately 30 to 60 gigabases of bases of DNA in a single run. The read length now is, I think, 250. This is still 2 times 150 base pairs, so which is quite some amount of data that we can generate. And the other machine that we very often use for bacterial genomes is the PacBio machine. The big advantage of this machine is the long read length, so that allows you actually to give a base for Illumina sequences, for Illumina reads, to assemble a complete genome. And we do the complete service from transcriptomics, genomics, genotyping, epigenomics, up to the bioinformatics. So what you get from us is a complete analysis. So if you produce this amount of data, you cannot leave alone your customers with that. So what you can deliver to them is only processed data. And to do that, we have quite some hardware. We have a 164 CPU cluster. We have more than 700 gigabytes of RAM, and of course storage like hell. And with this equipment, we are able to assemble complete genomes, transcriptomes, and so on. And we use different programs so that the list is filled with slides. And of course, for every step in this data management and analysis, we have a certain and tested set of programs that are in part self-generated, in part they are publicly available. And the last step mostly is a gene enrichment analysis. So we use gene ontology enrichment. You can do that with your own data set on our server. We have we provide KEC and BioCasa assignment and enrichment analysis, and of course gene set enrichment analysis mostly for human and mouse data. So why I'm here today is actually because I want to talk about drought research in chickpea. And as you can see, drought this is one of the slides from ICTSAT, from Manish Murthiwal, from a poster that came into my hands, and I like it very much. It's very clear. Actually, drought is the major constraint to chickpea yield improvement, and so ICTSAT has done a lot of work to tackle this constraint, and we would like to contribute to this effort by adding TranSNiPtomics. This simultaneous analysis of gene expression and of polymorphism. So what we can see using this technology is actually allele-specific gene expression. And the advantages of these technologies is that you get markers, SNPs and indels that are located. Can you hear me? If you cannot hear me, you tell me. Can you hear me? Yeah? So I don't need that. Can speak a little bit louder. Okay, sorry. Yeah. Okay, next time. Okay. Yeah, so what is the advantage of this technology? Actually, you can identify markers that are within genes. And that is the advantage is that these markers are very likely to connect it to a specific trait. So they may underlie the trait. And these markers can be chosen from differentially expressed genes. So you have gene expression, differences, and you have allele-specific expression. And of course, if you have this, you can connect that to a trait very easily. Before I go into detail now with transcriptomics, I just would like to remind you to a certain feature of the transcript. You know, unlike the DNA, the gene expression is very much depending on the gene that is expressed. So there are many genes that are expressed only in less than 10 copies per million. So this is more than 50% of all the genes are very lowly expressed. There's another part of genes, and this is actually less than 0.2% that make up more than 40% of transcripts. So you have a very large divergence of gene expression in the transcriptome, and of course this is very different from the DNA where you have one gene at a time, at the genome. And this poses certain problems. And this is actually a technology, RNA-seq, that is usually used to sequence such a transcriptome. RNA-seq dissects cDNA into small pieces, and these pieces are then sequenced. And that means that you have different lengths of transcripts, giving different amounts of sequences. And also, if you have a very abundant transcript, you get a large number of reads. So that means if you have a long transcript that is abundant, you get many, many reads of this particular transcript, but it does not help you in the quantification of the amount of the transcript. So, and that means that these reads vary per transcript and depending on the transcript length. And the problem is that it is very, very difficult in non-model organisms to assign a certain read to a certain transcript if you do not have a genomic sequence. And that means that if you want to have a good quantification of your transcriptome, you need to do a very deep sequencing. Especially if you want to sequence to see rare transcripts like those from transcription factors, kinases, and so on. So some that may have very, very important effects on other genes. So what we do instead of RNA-seq, we do massive analysis of cDNA. And that means that in this case, only the 3' ends are sequenced. And that means that we have a reduced complexity of the transcriptome. We do not see too many transcript variants. We do not see, for instance, alternative splicing. But we concentrate on the most polymorphic part of the gene, which is the 3' end. And on the other hand, it is not only the most polymorphic one, it is also very specific for a certain gene, much more specific than, for instance, an exon. And so you can distinguish members of gene family by their 3' ends. So, and that means on the other hand, because we are only doing one sequence per transcript, it is easy to quantify. And that means on the other hand that you get a very good, because you concentrate on one part of the genome, this is most polymorphic, you get enough coverage to get a good SNP detection. And that means it reduces costs, and that makes it useful for breeding. So, and that means that we can actually, using this technology, we can now analyze hundreds of genotypes for a reasonable price. And that means that we can, for instance, tackle complete populations. And this is actually how it works. So you have the cDNA here with a poly A tail, and we bind to this poly A tail an oligo-DT primer that is used for cDNA synthesis. This oligo-DT primer is connected to a streptavidin, to a magnetic bead. This magnetic bead binds then to streptavidin beads, and that means that this complete cDNA can be removed from the solution using this biotin streptavidin connection. Now we fragment the cDNA, and then we just, using this streptavidin magnetic bead connection, extract the distal 3' end from the solution. So what is remaining is just the region at the 3' end of the cDNA, and we sequence just into this 3' end. And so we assemble then these sequences. So you see here we have 1, 3, 4 assembled. Here we have only one read, and what we get is contigs that are 50 to 400 base pair long, and these are just counted. So how many reads were going into one contig? And you see in this case we have 4, in this case we have 1. So we have only one fragment per transcript. That means these 4 represent 4 different transcripts. So now you have these contigs. These contigs are now annotated or mapped to a genome or annotated to a transcriptome. And so you can now assign them to a certain function. You can say, okay, this is gene 1, gene 2, or even unknown transcripts. And so after the assembly, if it is an unknown transcript, of course you want to know what it is. And so you assemble that to a contig. This contig is then blasted against, for instance, a protein database like SwissProt or EMBL database, and then, of course, you get a function. And at the same time, you quantify the transcript, and then you can do, together with the analysis, with the assignment to a certain protein, you can do an enrichment analysis, and you can do that on our server with using this MACE2Go tool. And this gives you a scatter plot, for instance, like this one. And the red, for instance, these red dots here, they are transcripts that are differentially expressed and belong to a certain gene ontology category. And you can do all types of analysis starting from this point. I do not want to go further into that. Just to compare now this RNA-seq approach, where you do this many reads per transcript, to the MACE approach, where you have only one transcript, one read, and that means that you have, in order to get a good quantification, you sequence 10 to 20 times less than with RNA-seq. So this gives you a very good power for SNP detection. Because you focus on the 3' end, you get many reads for an SNP. So that is confirmed. Whereas if you do RNA-seq, you get only a few reads per transcript, per SNP. And that means you do not never know whether this is really an SNP or it's just a sequencing error. And the problem is that these new technologies, they produce many errors. So in order to be sure, you need a good coverage, and this is provided by MACE. So in this case, for instance, you would not be sure whether this is really an SNP or an error, whereas here you are sure. And this is how it looks. For instance, this is from a read transcript on, with approximately 160 transcripts per million. And you see here the SNP very clearly with MACE. We have done 20 million reads here for this library. And now if you look at the same region of the transcript done by RNA-seq, you see that you will get much less coverage, and the coverage here is too low to be sure that you have really an SNP or a sequencing error. Now coming to chickpea and to the IGSCT project that Rajiv mentioned before. Involved in this effort is actually Rajiv here from ICTISAT, together with his people. Another industry partner from India that did not show up until now. Günther Kahl from Frankfurt University, and my company also from Frankfurt. And the aim actually of this project is to understand the impact and the mode of action of a major QTL for drought tolerance that has been identified here in chickpea in the variety ICC 4958 by ICTISAT. And we want to know actually what impact does that QTL have on the molecular level in different genetic backgrounds. Of course, we want to identify the genes that underlie this QTL. We have to, in the frame of the project, we have to produce 2 RT-PCR markers for the transfer of this gene in this gene or genes in breeding, and if possible, to produce transgenes to prove the function of this gene. And this is actually the QTL that you have seen probably many times, identified not only in the population ICC 4958 times ICC 8082, that is the population that we are working with in the project, but also in many other populations, as Rajiv mentioned. Also, this is taken from a postdoc from ICTISAT. And so the material we are working on here is, of course, the ICC 4958 as the donor of the tolerance, carrying this QTL. This is actually crossed on one hand to ICC 8082, which is a drought-sensitive variety, and on the other hand to GG11, which is a moderately drought-tolerant allele Indian variety that I think is produced throughout the country. And so 2 breeding strategies were used here. On one hand, this offspring here between this cross was backcrossed with GG11, and the outcome is a nearly isogenic line that contains by marker-assisted backcrossing, and the outcome is a nearly isogenic line that contains this QTL from ICC 4958. And the other strategy was to produce an F2 and then recombinant inbred line. Am I right? Yeah. Yeah. Okay. Recombinant inbred lines that either they are tolerant or susceptible for drought, and we assume that the tolerant ones have this QTL. Now, this is actually what we did. We produced 12 chickpea libraries from this, So well watered in this case means well watered, VWW, or 04 means after 4 days of drought. RNA was collected and MACE libraries were produced from these 12 situations. Now the libraries were produced and the reads were actually annotated to this complete set here of different databases. Some we have generated ourselves, for instance, like the chickpea transcriptome assembly that we have done at GenXPro using all the publicly available information. That's been done by Luis Bellarmino in our lab. Then there are several databases that are publicly available. All of them we have used to annotate the MACE reads that we have generated for these 12 libraries. Now, the first result was that we see that ICC 4958 regulates up a lot of different transcripts, and ICC 8082 regulates down a lot of different transcripts. And the same is true for the susceptible RILs. Also they regulate up many different transcripts, whereas DG11 regulates down many different transcripts. Again, DG11 plus regulates up many different transcripts, but not so the recombinant inverse lines. They are much more lazy. So somehow they are lazy and still tolerant. Now, and this is actually how it looks. There is something that we would say we call priming. And what you see here is a clustered heat map that relates actually the different transcription profiles and produces a similarity tree. And you see that in this whole transcription profile, ICC 4958 clusters, not as well watered without stress, already with these stress varieties. And so we call that priming. That means that there are mechanisms underway already in the ICC 4958 that are only upregulated in the other varieties under stress. And looking at these transcription profiles, you can actually see the breeding history here. So you see here, this is a gene ontology annotation of the transcript and enrichment analysis, and you see each category here is a gene ontology category. And you see, for instance, that here the most responsive category in ICC 4958 is respiratory electron transport chain gene ontology category. The same is true for the ICC GG11 plus. So, but if you look at GG11 here, you see that none of these categories is enriched in these drought stress GG11 plus. So probably this reaction, this is what we see. The reaction to drought was probably transferred from ICC 4958 to GG11 plus, probably by the presence of this 2KL. So this 2KL has a lot of different effects on the behavior of this GG11 plus. And now if we look to the tolerant RRL, then we see also this tolerant RRL has obtained a lot of categories enriched from ICC 4958. And this is less so true for the susceptible RRL. So already here we learn what is important actually for becoming drought tolerant. So these are mostly categories that are somehow concerned with mitochondrial function. So obviously mitochondria are very important components for getting drought tolerant. Now, and if you look then to the susceptible parent or to the susceptible parent, like the GG11, they are not activating these very important drought-tolerant function. So, as I said, these are mostly like respiratory electron transport chain, NADH, dihydrogenase activity. All these are mitochondrial reactions that are enriched in this nearly isogenic lines that probably got this QTL from the ICC 4958. Now, if you look into, we have a closer look into the recombinant inbred lines derived from the course ICC 4958 times, this is 8082. We took a bulk segregant approach. So we took 9 tolerant cells and 9 susceptible RILs, and we produced MACE libraries from these, and then we annotated these reads that we got, approximately 3 to 10 million, that are really useful to the transcriptomes that I showed before. And you see that in the tolerant bulk, for instance, only approximately 60% can be annotated, which means that the database is highly does not contain all the transcripts that are important for drought tolerance. So we need to improve this database. This is not so true, for instance, for the susceptible bulk. You see more than 80% of the reads can be annotated to the database that we use. And now if we look into the gene expression, we see that under well-watered conditions, there's a difference already in the bulk of approximately 400 genes that are differentially expressed between the tolerant and the susceptible bulk. And this is a very strong p-value. So if you lower the p-value, that might get less stringent, then probably this goes up several times. And then if you look into the reaction of the under drought, then we see that again approximately 500 genes are differentially expressed and more upregulated in the susceptible in comparison to the tolerant than downregulated. And of course, the reaction of the plant itself, the same bulk, approximately 1,000 genes show this very stringent differential expression, more in the susceptible varieties than in the tolerant varieties. So susceptible varieties need to do more to become drought tolerant. And this is actually how it looks, a scatter plot as I showed before. So here under well-watered conditions and here under stress conditions, these genes are upregulated under well-watered conditions in the susceptible RRL, and these are upregulated in the stress conditions in the susceptible RRL. And you see much less are upregulated or downregulated in the tolerant RRL. So you see already from the scatter plot the differential reaction. And this is now if you compare the susceptible and the tolerant reaction to stress. And you see then a bunch of genes that is only expressed in the tolerant bulk. And of course, these are very interesting. You must ask what this is. Again, as stress, these are mitochondrial functions that are upregulated and that are enriched in the tolerant varieties. And this is in special oxidoreductase activity, acting on NADH and NADPH, electron transport chain, as I shown, all upregulated or enriched in the tolerant varieties, whereas the response to stress as a category is almost similarly expressed in tolerant and susceptible bulk. So, and we are not the first one to see that these mitochondria are important for stress responses. This is a paper by Atkin and Macharel from 2009 that shows the same. So, of course, these transcripts that are and alleles that are unique in the tolerant RRS are, of course, potentially powerful markers for drought resistance. So, and we found actually 58 transcripts that were exclusively expressed in the tolerant bulk with more than 50 copies under stress and well-watered conditions. The priming that I said before, they are also already expressed under well-watered conditions and not expressed in the tolerant bulk even under drought. These are, for example, heat shock proteins, LIA proteins, and especially many unknown transcripts that have never been seen in the context of drought. So there's something new in these data sets also. Now the question is, of course, how are the alleles distributed between the susceptible and the tolerant bulk? And so we look now into the allele distribution and the transfer of alleles from one parent to the susceptible and the tolerant bulk. And just to give you we have an automated workflow for non-model as well as for model organisms to identify, these transcripts, and what you get out is a table like that, as an Excel sheet that gives you the reference base, the variant base, the number of reads in the reference base, where the reference base is seen, is sequenced, and the number of times the variant base is sequenced. And then it gives you a 100 base pair before and after the SNP that gives you a good opportunity to assign timers for PCR assays and so on. So, and this is how it looks in chickpea. We have an unknown transcript from chickpea, and you see actually, and it's not annotated, and you see the SNP here, a T in this position, and a C at the same position in the other allele. And we have enough coverage here to be sure that this is really true. This is not a sequencing error. So just to summarize, we have detected alleles in parental lines and tolerant and susceptible bulk, approximately 4,000 that are exclusively found in either one parent, so these are exclusive SNPs, and approximately 130 that are at least 10 times in both parents. So you have the allele A and B in parent A and parent B. So if you look into the bulk, then we find approximately 1,250 alleles that are exclusively found either in either one bulk, so that this differentiates between the bulk, and those that are really interesting because they are contrasting, they are exclusively alleles expressed at least 10 times. It's important to be sure it's this one in both bulks, and this we get 12 of these. Now, where these, if you look into the distribution in a general way, then we see in the resistant bulk got approximately 85% of all the alleles that are overrepresented from ICC 495E, whereas approximately 84% of the susceptible got these specific alleles that are overrepresented from ICC 1882. And the green one here are the alleles that are there in equal numbers. So you see that there is a unequal distribution, a very unequal distribution of transcripts going to the susceptible or to the tolerant or to the susceptible varieties from the different parental lines. Now, if you look into the QTL region, you see here a sketch of the QTL region together with the markers that has been used for marker assisted backcrossing. So you have a bunch of genes in this region. I think at least 20 that we have identified, maybe more. And so what we are looking here in these genes are exclusively to the 3' UTR, to the non-coding region at the 3' end. And these are the SNPs that we are looking at. So they are non-coding SNPs in most cases. And then we look into the distribution of these alleles in genes from this region. And what we get out then is how often a certain base has been identified in the different genotypes. So, for instance, this blue one is a G in position 322 or a T in 322, position 322. And then we have a look how much this is occurring in the different genotypes. And for instance, we have a ninja family protein here in this region, ranging in this is the genomic position here of this particular gene. And you see here that we have a polymorphism here that occurs in ICT4958, we have a G, in ICT8082 we have a T, and we have that across the whole gene 3 times, so we have 3 SNPs here in this ninja protein gene in this QTL region. And the same is true for instance, for here this time for coffee-like, to the end of the time for coffee-like gene. Time for coffee includes the nuclear regulator in the Arabidopsis thaliana circadian clock. So this is a probably a circadian clock gene, and you see here that this G, which is present in ICT4958, also occurs in GD11, but also in the tolerant variety, and that instead the A in this position 1976 occurs mostly in the susceptible line. So also the susceptible RL got this A from 8082. So in summary, we have found in the 166 kilobase per region in this in this QTL, we found 11 genes that contain 30 SNPs. These are listed here. Sometimes 6 in one gene down to only 1. So I think this is a nice source of SNPs coming directly from genes that are expressed and may play a role also in drought tolerance. And what we are doing in the moment, we are extending this analysis from bulk segregant analysis to the whole population. So we have 75 RLs or 75 MACE libraries, I should say, from this box that I mentioned before, and we have done the sequencing so far. We have from the tolerant RLs, we have approximately 360 million reads. From the susceptible RLs, approximately 270 million reads, a total of 660 million reads, and of course also several million reads from the parental line. And so what we are going to do with that is actually eQTL analysis of this recombinant line population. So to just summarize, the gene ontology enrichment analysis of this in both cases, in the recombinant inbred line population as well as in the nearly allogenic line, indicates that there is a strong involvement of mitochondrial function in drought tolerance of these lines. We have identified a whole set of highly reliable SNPs in coding regions. We have shown that MACE is really a cost-efficient technology that allows you to simultaneously identify differential gene expression and allele-specific gene expression. And so we have just started to do this, to apply this technology to recombinant inbred line population, and if you are lucky, we will manage to get a MACE without chickpea. And these are actually the people that are involved. From the ICTSAT side, of course, Bindu Udapa, who was involved in senders the RNA and just rotated because so many times it was not useful. Thank you for your patience. And Ajiv, of course. In my lab, Nicolas Petschorn did the bioinformatics. Björn Rotter was the head of the laboratory. Camilla did the part of the bioinformatics analysis. Anja Frank was responsible for the MACE library. And I did the graphics. And these are the people involved from Jutta Karl's lab, Jutta Koll, and Nicolas Gonzalez, and the whole thing was paid by the Indo-German program of the BMBF. And I thank you for your patience.
Thank you very much. This is not chickpea flour, right? No. This is different. Okay, good. Anyways, I mean, this was a great presentation, and just to add that Peter mentioned that several other colleagues were involved, so Hema was not just involved in that sending the RNA, but also together with LK, with Mahendra, in setting up the physiological experiments, which are very challenging. And then, of course, once we have these data, then, and of course with Puran Gaur, and then once you have this data, then these people together with Amir was involved, or in analysis, etc., many people, but we can discuss those things later. One thing, as you could see from this presentation, and this is always important, to have the good genetic material. And now we could figure it out, such kind of mitochondrial related genes, or their involvement of the energy related genes, for providing the drought tolerance only when we have the JG11+, which was really done, of course, by Puran Gaur, by interrogation this QTL. So point what I'm trying to make, that just genomic, just sequencing technology, they are not going to be helpful unless you have the good genetic material, you have the good phenotyping data, you are having the good information, then only you can make some sense. So this was really good. And as Peter tried to give an idea, and again during lunch we were either can say gossiping or discussing, we were talking that, well, this QTL region we are trying to dissect by using different methods. So one of our PhD student, Deepa, who is sitting in the corner, so she is involved by using the different approaches, and then together with Peter, so we are using this approach, MACE library, together with colleagues from ACPFG, Tim Sutton and Dave Edwards from University of Queensland, we are trying to enrich that region. We are sequencing the recombinants. Now we are also using the QTL-seq approach, so different thing, but good thing is that now we got the good stuff where you can enhance the genomics research, you can enhance your basic understanding, so this is good. Anyway, so now we can have a few questions related to this talk first, and then if you would like to have some words of wisdom from Peter, then he will also be happy to give that. So let's first have the questions related to talk, right? Yeah. Good. Yes, Dr. Gaur.
Good presentation, Peter, as always. I just, one of the things in all these transcriptomic analysis is that, we always end up in having large number of genes upregulated, downregulated. Now, can we, unless we go further to see the effect of individual genes, in terms of values, that they contribute to either in enhancing or declining, you can't, decide that, which are the one which to be followed up. And in this particular case, we are curious that, of course, we got the QTL region, and if we interrogate the whole, we get this much advantage. But if we move beyond that, to see the other genes, how much benefit we are going to get, unless we have some assessment. It's very difficult for a practical breeding program to how to, use this. So just your thinking on how we go further down there.
Yeah, thank you for that. This is actually the idea behind this unit, to go to the complete population. So imagine you have approximately hard to measure 1,000 quantitative on the morphology. The second thing that you get at the same time is a very dense image set. You remember I thought that there are approximately 4,000 LNPs that segregate in this population from this different two forms. So what you get, if you analyze the whole population, then it's not, unfortunately it's not that big. You see here the sequence, but you do not see the complete analysis. So what you see is actually 4,000 LNPs, yeah, 4,000 LNPs and 1,000 quantitative specs. So put that on a map and you will see exactly the contribution of every local and every gene expression to your space. The question is, so that means that the map is very wrong, has been shown by any knowledge. But you get to the very basic map, and at the same time, you get not only an FNT map, but you get an issue map. And this will directly run you into the field. Not when you think these people are expressed or not. If you look into the genomic system, the FNT is there, but you do not know whether it is expressed or not. And you see that very often, for instance, in cancer, if you have FNT, but there is no gene expressed. So probably this FNT does not have any context for the trace. And so this is, I think, is a good path to put that.
Yeah. Very good. Good. They will tell you later, Peter. Okay. And other questions? Yes, Gaurav. Come close to that mic and then can speak. You can sit here also, yeah.
About the aspects of SNP and expression business. Why aren't we focusing much upon the SNPs which can be identified from the promoter region of gene itself? Because the promoter is the one which guides the expression of the gene. So wouldn't it be a good idea if we focus on the promoter of a gene and figure out the SNPs from there? So because ultimately the transcription factor is going to bind on the promoter. And if there is an SNP in the core promoter region where the transcription factor binds, and which might be leading to the improper binding of the transcription factor, which might result in differential gene expression. So that is one aspect that I intend to know because my knowledge is limited in this area.
I agree completely. So the best thing would always be to measure everything. And we discussed this afternoon, we discussed the influence of epigenetics. So it may well be, that there is no SNP in your promoter, but it is just epigenetically silenced. Yeah, so it would be great to have the information. The question is why it is epigenetically silenced, and one of the reasons could be that there is a small RNA directing a methylase to this place. And so what you would see is the differential expression. If you look into the small RNAs, we do that regularly, and we also look into the epigenetics. Then you see a small RNA that is supposed to bind to this promoter region, and then it silences the promoter. And what you see is the promoter differentially methylated, and the small RNA differentially expressed. Of course, this would be great. But what is the effect of that? The effect of that is that a transcript is less or more abundant. Another factor that influences the abundance of a transcript is the availability of microRNAs. Yeah? So if you switch the availability of microRNAs, then you switch whole sets of genes. That's one of the things that we should think of and have to do. Yeah, okay, yeah. So I could give you a lot of material about that. This is actually what we frequently see, especially in cancer research, where you have all the information. It's a little bit behind that in plants, but also emerging, very well explored investigated in Arabidopsis. So that is another aspect. So that is not just the SNP in the promoter region. So there are many effectors that regulate gene expression. And the question is, what is the most meaningful to measure? And our answer was, for this type of work, the effect, everything is expresses itself as gene expression. No, that in the end, if the transcript is there, is another level of regulation, but it is not so strong as the microRNA regulation. So my answer is, of course, it makes sense to measure all these, to understand more, but for practical reasons, the transcription and the SNPs. So where is the expression coming from? Which allele is transcribed? Are the most meaningful things to measure. Ja, alles klar. You were almost done.
Okay. So any other question from our gene expression ladies? Okay, Babu will ask first question, then yeah, you can come here.
Yeah, actually we can see that there are a number of positive things from this approach. One of the things I'm wondering is, in case if we have some genomic variations within a transcription factor or an upstream gene, which do not result in expression difference per se of the gene, but then it leads to a number of downstream gene expression differences. So are we running a risk of not being able to identify them in this approach, or is there a way to get to that?
The answer is yes. So you cannot expect from this type of analysis to give you all the answers. So for instance, there may be, if there is no differential gene expression in the transcription factor, the question is what this transcription factor then contributes to the differential expression of other genes. And one of the reasons could be, for instance, that it is that there's an SNP that we do not detect using this technology. No? So we only look at 3' ends. That means that we detect approximately 30 to 40% of the complete allelic diversity that is in these transcripts. So you do not see everything. Another reason could be, that this transcription factor is, for instance, differentially phosphorylated. Yeah, and so we do not know why. That could be on another chromosome, in the kinase that is just, and this kinase is differentially expressed and then activates transcription factor. So we do not measure phosphorylation here. Yeah, so there are many aspects that you cannot measure with this technology, and that's the reason why so many techniques are necessary actually to understand everything. The question is what you can use in a broad effort. Yeah, so, and this is because of next generation sequencing, otherwise this would not be possible. This can be done now on a large scale, and it is relatively cost efficient. But of course it cannot do everything.
Okay, good. Last question from Mike, please.
Yeah. I mean, the question is how complex is a trait? So what is the influence of a single allele or a single gene on a trait? I mean, what is absolutely clear, for instance, that the QTL that has been identified here is not the only player, but it is a major player. And you know yourself that you can have influences on traits ranging from 0.1 or 0.1 to 80%. And so the question, so that means that it depends, of course, on the size of the population that you are looking at, so the resolution that you can have, and on the other hand of the influence of a single component on the trait. So if you have a very strong component, you will easily identify this component. If it is a weak component, you get a small buckle in your probability curve, and that may change actually if you go to another population. You know, it may change its position. You know, it becomes the most important under other circumstances. So this gene interaction is very important, and everything that you can say is, of course, true for the population that you are working in. If you go to another population, things may change, and you can only hope that the component you are looking at is also important in the other population. And I think for this QTL, ICRISAT has shown that this is important not only in the population ICC 4958 times 8082 that we have analyzed here, but also in other populations. So this is a valid stuff. It may change if you go to minor QTL.
Good, very good. So I think that we had very good presentation and then subsequently good discussions on different aspects. So any other question, or I think we can close? No question, right? Okay, good. So at this, now I would like to avail this opportunity to thank Peter for wonderful presentation and for extending a great collaboration to ICRISAT. So please join me in thanking once again to Peter.